Okay, good afternoon. It's a pleasure to be here, and I really thank SBIA for inviting me to present here. My name is Cecilia Tami, and I am from the Office of Biotech Products. We are going to discuss today the review of chemistry, manufacturing, and controls of investigational new drug applications. So this is going to be a shared presentation. I'm going to start, and then Bala is going to continue, and then we come back together to answer any questions that you may have. So just to go through what I'm going to talk about, first I'm going to do a brief description of the structure of the Office of Pharmaceutical Quality, OPQ, and then explain to you why the CMC information is relevant for an IND and, of course, for a BLA or NDA. Discuss a little bit what are the differences between small molecules and biologics, and then dive into the CMC information that is required for an IND. I will specifically go into details for the drug substance, and then Bala is going to elaborate on drug product, and then the path forward for NDA and BLA. And I am going to share some case studies to show how we identified CMC safety concerns and how we resolve them, and work together with you to resolve them. So this slide shows you the structure of the Office of Pharmaceutical Quality, or OPQ. This office was created within CDER as a single unit dedicated to product quality. And it stood up in 2015, and it covers all the functions related to quality. That is review, research, inspections, life cycle, surveillance, policy. And the idea is to have one quality voice and have a better overview, an improved overview of all, of quality of the product throughout its life cycle. It consists of 8 sub-offices, and the CMC part of INDs, BLAs, NDAs is reviewed in the Office of Biotech Products and in the Office of New Drug Products. I'm from the Office of Biotech Products. Bala is from ONDP. And so this new structure at CDER ensures that the quality of medicines are available for American public. So why is it that product quality is important? So during your IND, during development, you manufacture lots. And you use these lots, these are your clinical lots. You use these lots in clinical studies. So your clinical lots have a quality profile, and then when you use them in the clinical studies, you collect safety and efficacy data. But once your product is approved and is in the market, so how do you ensure that your commercial lots are going to have the same clinical performance that your clinical lots? And this is through ensuring that your manufacturing process is under control and it's able to deliver a product of consistent quality every time you manufacture. And so basically the quality is the anchor. Manufacture and this is kind of summarizing what I have just said, manufacture of a product of consistent quality assures the clinical performance of commercial lots with regard to safety and efficacy to be the same than that of the clinical lots. So both small molecules and biologics are drugs because they are used for the treatment, cure, and prevention of diseases in human. Biologics are a subset of drugs. At CDER, we regulate or review some of them. Some others are at CBER. So what are the differences between small molecules and biologics? The most obvious one is the size. So small molecules are small, and biologics are generally proteins bigger than 40 amino acids. Together with the size comes the complexity. And generally, biologics are more complex and manufactured in living material, while small molecules are synthetic or semi-synthetic. The structure is more complex for biologics. It's better defined in small molecules. And both small molecules and biologics are regulated under the Food, Drug, and Cosmetic Act, but also biologics are also licensed through the PHS Act. And because of that, there is a requirement to demonstrate potency of a biologic, while for a small molecule, you need to demonstrate strength in addition to purity and safety. Small molecules are reviewed at ONDP, and those biologics at CEDAR are reviewed, most of them at OBP. So this slide's a little bit to elaborate on what I have just said. So you can see, let me go back so I do the presentation timely. So this is statin. This is a small molecule that is used to lower lipids and prevent heart attack and stroke. And this is a monoclonal antibody. As I said, it's obvious. Statin, as a small molecule, is small. A monoclonal antibody is bigger. And it's more complex, and that also can be, it's clear from the image. So what is also important in biologics is that the structure is very important to assure the biological activity. And because the manufacturing process is complex, small changes in the manufacturing process can have huge impact on the quality, the structure, the activity, the purity of the product. The other safety concern that comes with biologics and some small molecules is that when they are manufactured in living cells, then they have the potential to transmit infectious diseases. This slide shows a non-exhaustive list of the class of molecules that are reviewed in ONDP and in OBP. I just want to make a mention of the last line. So there are some that refers to antibody-drug conjugates. So there are some drugs that when they have a biologic component and a drug component, we both do a part of the review for the same product. How do we review the IND at FDA? So we do it like as required by the CFR, this is 31222. Basically, the objective is to assure the safety and right of subjects for all phases of development. And then for phase 2 and for phase 3, in addition to safety, we need to evaluate efficacy. And there are some guidances that elaborate on this concept, like this one that I show here. Before you submit your IND, you can request a pre-IND meeting to discuss the readiness of your submission. From a CMC standpoint, the idea is to discuss all those aspects as relate to identity, strength, quality that could pose a concern. What is the information that you might need that you don't have, that you might need to provide in the IND to make sure that is a go? So what is the CMC information that is required in an IND? This is again described in the CFRs and includes a description of the composition, manufacture, and control of your drug substance and your drug product. What is it? How do you do it? Where you do it? And then sufficient information to ensure that its quality, purity, and strength. The amount of information that we will expect is dependent on the phase of development. At phase 1, you may have less information, and as you progress, you will understand your product and your process more, and so you will be able to submit much more information to us. So to go into more details, the CMC package of an IND should have information about the drug substance, about the drug product, the placebo, the labelings, should have an environmental assessment, and also some kind of small CMC description in the investigator's brochure. Well, so now I'm going to describe a little bit more in detail what is the information that we expect for drug substance. Let's start with the definition. And drug substance is an active ingredient that is intended to furnish pharmacological activity or other effects, and is not to be used in human subjects. What is the information that we expect for drug substance in your IND? We need to understand what are the physical, chemical, and biological characteristics of your drug substance, where and who manufactured it, and how you do that, how you control for the characteristics of your drug substance, what are the methods you use, what are the limits you are going to use. And then information to support that your product is going to remain stable during the duration, well, for the duration of the clinical studies. This slide shows you a typical, or a manufacturing process, mostly for a biotech product. You will have an upstream process and a downstream process. During the upstream process, you will start with your cell bank, in the case of products that are manufactured through living cells. And then you're going to amplify those cells until you get to an amount of cells that are able to provide you with the amount of product that you want to produce. So that is part of the upstream process. Whether your product is expressed inside the cells or secreted to the supernatant, and then you are going to collect the cells or the supernatant, and that is going to be the starting point for the downstream processing. The downstream processing then are a series of steps, purification and concentration steps, until you get a product with the expected purity and quality. To have your manufacturing process in place, before that you need a cell bank, and all the information about how you generated that cell bank, which was the parental cell that you used, what was the vector, what is the sequence you transfected or transformed, how did you clone it, all that information should be provided in the IND. And for most of the biotech products, this starting material is animal or is of biological origin. And therefore, like mammalian cell, bacteria, yeast, there are some small molecules that are also manufactured from living material. So all this material, as I mentioned before, are potential sources of adventitious agents. Here you have bacteria, fungi, viruses. And not only that, but also an optimal environment for the growth of adventitious agents that may go into or come into the process through other sources, raw materials, environment. So because of that, it's very important that the cell bank is tested. And it's tested for not only for identity, but also for purity, and when appropriate for the absence of adventitious agents, either not endogenous or endogenous. Because the presence of adventitious agents is a safety concern, this testing should be performed and the information provided to us. So as I mentioned, the cell substrate could have the potential to harbor viruses, but also viruses can be introduced into the manufacturing process through raw materials of animal origin or biological origin, and also during manufacturing, because the water is contaminated, the equipment might be contaminated, the excipients. So again, because the potential presence of adventitious agents in the process or in your product is a safety concern, for a phase 1 IND, you need to support the viral safety of your product. And this has different layers. The first one, as I mentioned, is to adequately characterize the cell bank. The second one is to have adequate control over the raw materials of animal or biological origin that are used in your manufacturing process. You are expected to test the unprocessed bulk, that is the harvest, the starting material for your downstream process, and that is because that is the material that is more sensitive for detection of viruses if they were present. And then you should provide information demonstrating that your manufacturing process is able of removing or inactivating viruses were they there. So, and the expectation for a phase 1 IND is that there is information for at least two orthogonal steps in the manufacturing process for their capability to inactivate or remove viruses. So how are these studies? So you need to demonstrate that your process is able to remove viruses if they were introduced into the manufacturing process. So how do you do this demonstration? And generally, and this is done in small scale studies. It's not done at full scale. It's not done in your facility because you don't want to introduce viruses in your manufacturing place. So generally, these studies are contracted out. So there should be small models that are representative of the full-scale step that are used to demonstrate the capability of that step to remove or inactivate viruses. And basically, these are spiking studies where you spike high-titers virus, you pass them into intermediate material, and then you pass it through this small scale, and you measure the amount of virus that is in the recovered material. And this difference is going to tell you how many logs, hopefully many, this step is going to remove or inactivate. So as I mentioned, they have to mimic the purification step for a phase 1 IND. Again, we expect two orthogonal robust methods. And we expect a summary information that justifies the adequacy of the small scale to represent the full scale. So we need data to really determine that the results that you got in the small scale apply to the full scale. And so this information is used to calculate the log reduction factors, how much virus you have at the beginning, how much you have at the end of the process, and then this is also used to calculate the safety factor. So this is a case study that in which the sponsor submitted an original IND for a recombinant protein in CHO. So they provided data to support the viral clearance of 2 steps, orthogonal steps, and then when they calculated the log reduction factor, how many logs of viruses could each step remove, this was quite low. So the capability of these steps to remove virus was quite weak. And then the submission did not have information to support the adequacy of the small scale model to represent the full scale model. So they were placed in clinical hold. And so when in their response to hold, the sponsor reassessed these 2 steps and included and claimed another, a 3rd step for viral clearance. So then they could achieve a higher low reduction virus, and provided data to support the adequacy of the small scale. And so this IND was allowed to proceed. So just to wrap up, to support viral safety is very important for a phase 1 IND. This includes characterization of your cell bank, testing of unprocessed bulk, control over the raw materials of animal or biological origin, and demonstration of the capability of your process to remove viruses through 2 steps that should be robust and orthogonal. The IND should also have information about the characteristics of the drug substance, including physicochemical characteristics, identity, primary, secondary structure, potency, or content, immunochemical properties, quantity, purity, impurities, contaminants. We need to understand what is the purity and the profile of your product, and what are those impurities, whether they are process-related or product-related. And for all these, to support all this information, we expect not only tabular data, we don't expect only numbers in the table, but we want to see the primary data, images of the gels, chromatograms, something that allow us to make our own assessment. So this slide is not for you to read every single word, but just to give you an idea that there is a huge variety of methods that could be used to characterize your drug substance. And some methods are more suitable for small molecules, others are more suitable for biologics. But in any case, there should be methods to assess the safety from an endotoxin and bioburden standpoint, methods to describe the purity and the impurity profile of your product, identity, potency, and protein content. From this whole big number of assays that you may use for characterization, some of those should be selected as release assays. And these are assays that are going to be used to confirm the quality of your drug substance. So you know a lot about it, but then you are going to identify what are the critical quality attributes of that drug substance that you want to control at release. So these should assure the quality, the safety, and the doses of future lots. So you establish specifications at release, and those specifications allow you to have assurance that the lot that you manufacture today and the lot that you are going to manufacture tomorrow are going to be comparable. When we refer to specifications, this includes the method and the acceptance criteria that you are going to test against. And again, as for most of the information that you gather during development, these specifications are going to be broad for a phase 1 IND because you have manufactured very few lots, or maybe 1 or 2. But then as you gather more and more information, you have more experience on the manufacturing process and on your product, then they are going to get narrower. So release testing should include a test for identity. That has to be a unique test for your product, small molecule or biologics. Test a set of tests that are going to characterize the purity and impurity profile of your product, including product-related and process-related impurities that might be present in your product. Potency, there should be specification for potency for biologics in the case of, and for small molecules, but in the case of biologics, these are, the potency test is a test to assess the biological activity of your product and should be reflective of the mechanism of action of the product. In the case of small molecules, is a determination of the content, and reverse phase HPLC is a very generally used. Specification should test for quantity and for microbial safety. But in addition to testing at your drug substance at release, you should test in process material, and because this is going to give you a better control of your manufacturing process and also monitor that each step of your process is performing as expected. So at points that you determine are important, you're going to test for different attributes, and you're going to establish alert or acceptance, or no acceptance, alert or action limits. And this control over the manufacturing process is going to assure that you get the product that you expect. CFR also requires that stability data are collected during all phases of the IND. And these data should be sufficient to support the stability of your material during the toxicology and clinical studies. Stability data should be collected under the recommended storage condition for your product. So if you plan to store your drug substance frozen at -70, you are going to collect data at -70. And but it's very important also to collect data under conditions that are under accelerated conditions or stress conditions, because these are going to give you a lot of information about, well, the degradation pathways of your product, and also help you identify what are those assays that you want to use to monitor the stability of your product. So these kind of studies under accelerated or stress conditions, and they are going to help you identify which are those assays that you're going to include in your stability testing. It's very important, or it's recommended, that you collect data as early as possible during their development, so to put the toxicology lot and the first clinical lot in stability, so that when you come for the IND, you have data to support that your product is going to be stable during the planned clinical studies. And then, of course, you keep on collecting data throughout development. So this case study refers to stability, obviously. So at the time of the original IND, the sponsor submitted stability data from the tox lot. They hadn't collected data for the clinical lot. But the problem was that the stability data for the tox lot showed that there was an increase in the impurities by reverse phase throughout 6 months when the tox lot was stored at the recommended storage conditions of -70. So really, we didn't have information to support the stability of the proposed clinical lot during the studies. So this IND was placed on hold. And this IND was removed from hold while the sponsor provided stability data from the clinical lot with the primary and tabular data supporting that the clinical lot remains stable. Well, you all know that phase 1 is safety, it's about safety. The reasons to place an IND of phase 1 IND on hold are safety concerns. And those could be that there is not sufficient information to assure the safety, to assess the risk to subject, or that the information does not ensure that humans are not exposed to unreasonable risk. So from a CMC perspective, the safety concerns could be due to the nature of the molecule. You may have a toxin, you may have something that is carcinogenic. So the nature of the product that you are developing in itself may have some safety concern that you need to address. A safety concern could be the presence of impurities in your product, process-related or product-related impurities that may pose a safety concern, immunogenicity, or maybe some toxicity. And then there are general safety concerns regarding bioburden or endotoxin, the potential presence of viruses. If you don't provide sufficient assurance of sterility, that is in the case of drug product, residual solvents, levels that are kind of unacceptable. So how do we ensure that your phase 1, your proposed clinical material is safe to be administered to humans? And so this safety evaluation is done based on the non-clinical study results. So you are going to, you generally have a tox lot, and you do your non-clinical studies. And so these studies are going to inform about the pharmacological effect, the toxicology effects of your drug. They are going to also provide some proof of concept of how is the mechanism of action and inform the initial safe dose. Now, you may use the same toxicology lot for the clinical studies. And in that case, the safety of that material has been already addressed because it was used in the tox studies. However, if your clinical material is different from your tox material, how do you ensure that this clinical material is safe? And this is done by establishing a comparability between the tox lot and the proposed clinical lot. I guess that in most of the cases, the tox lot is done under a non-GMP environment. So most of the sponsors, what we see is that the GMP lot, the clinical lot, is different from that lot used in the toxicology studies. So the question becomes whether the non-clinical material is representative of your proposed clinical material. So this representative is addressed by establishing a comparability between your tox material and your proposed clinical material. And this is risk-based. So what are the differences in the manufacturing process for your tox lot and your proposed clinical lot? What quality attributes could be impacted? And so all this information ultimately informs what are the kind of analytical comparability that you should perform. Again, when you submit this information to support the safety of your clinical lot based on comparability with your tox lot, we want to see tabular data, but we want to see primary data too. If you do it side by side, that is great. If not, it's acceptable. And again, this comparability should focus on those attributes that are related to safety. Process, impurities, potency. So in this case, we receive an IND for a recombinant protein. And the release results for the tox lot and for the clinical lot met all the release acceptance criteria. But the IND didn't have the primary data, so we requested that data. And when we looked, when we reviewed that data, we saw that there were differences in the purity profile of the clinical lot when compared to the tox lot. The clinical lot had some low and high molecular weight bands in the SDS gels that were absent in the tox lot. So this was a concern. And these bands were not detected by Western blot, what meant that they were not product related. So the IND was placed on hold because there was insufficient data to support the comparability between the tox and the clinical material. And so the sponsor provided information, explaining what was the nature of those bands and why they would not pose a safety concern to patients. Another potential safety concern is immunogenicity. For a phase 1 IND, we don't, unless the product is high risk, we don't expect that you have assays developed or validated. But the expectation is that you collect samples at appropriate time points in your clinical study and freeze them under conditions that are appropriate for them to be tested once you develop your assays. And these last two slides give a list of our more common CMC-related hold issues. I think that I went through most of them with the examples and during the talk. I think so. Okay. So to summarize, for a phase 1 IND, we expect sufficient information to assure viral safety, data to support the comparability of the toxicology lot and the clinical lot when appropriate, when they are different lots, adequate specification to ensure that the quality of the lots that we are seeing, because if your proposed clinical lot is going to be the same and the same and the same when you keep on manufacturing material. and data to support the stability of the product during the clinical studies. And with that, I give the floor with Bala. Thank you so much.
If you give me a second, I would like to take a sip of my favorite solvent. Thank you. It's really a privilege and a pleasure to be back at the SBI conference. I really do appreciate the opportunity to be here. Before I start, I just wanted to share a thought about it. As I was preparing for this presentation, I wondered if there was any one other conference that I had participated repeatedly with so much enthusiasm. I just couldn't find one. And there were several reasons for that. And one is that I could meet all my colleagues from the campus who I otherwise would not be able to see them, but also that we could come here and share our regulatory angle for some of the submissions we receive and we review. And most importantly, the format of this conference allows this interaction from 4 to 4:30. I'm really looking forward to it. I'm sure you too are. And that is something that I cherish because we can take back some of the experiences that you share with us, the questions you come up, and the issues that you discuss. So I really do appreciate that as well. And I thank you all for sending us some of the wonderful submissions, which is making our work very interesting, rewarding, as well as very gratifying. Thank you. So could we please go to the first polling question? And for those of you in the room, I just wanted to bring this up and talk about what is one term that you prefer using or you've been using in your developmental program. Is it investigational product, drug, study drug? Anyone who wants to volunteer to? As the poll is going on? That's quite a poll there. It's shifting. Okay. Since the title is for investigational new drug, I suppose investigational drug is the one that goes. So I just want to make sure that we are all on the same page because I would have used any of the above or none of the above or of the above or something different. Just so we are all on the same page that we make sure that we understand where we are. So it was a great presentation by Cecilia to talk about the drug substance, and I'm going to take about the drug product formulation and talk about the various subcategories within the drug product section. And I have a few slides that will walk you through what are the subcategories within the drug product section. And then I would have some case studies where we almost went to a clinical hold, and believe it or not, we don't want to put any IND on clinical hold because we really do appreciate what you do, and it's a time that's a huge factor, and that's something that we want to avoid at any cost. And one of the things that I want to make a mention here is the reviewers who take on a heavy workload with the NDAs, supplements, meeting packages, and also IND. So when an IND comes to them, they just stop working on other application, take on this to first figure out if there is anything that would trigger a hold. And I really do appreciate our regulatory colleagues here who get on the phone or send you an email saying, CMC wants these information. Could you please provide them so we can get this going and we could say it is safe to proceed, at least from a CMC point of view. And then finally, I'll touch upon where we take it from the 30-day safe to proceed. So we could have an IND amendments, annual reports, and then eventually what we want to see is an NDA and a BLA submission come in, and we work with you to make it a good product available for the cause of public health. Just by show of hands in the room, is there anyone from here who attended the spring session conference in Atlanta? Okay, so we have one. I'm all for green chemistry, so I'm going to recycle some of the chemistry jokes. So you are the catalyst to motivate the rest to join. Okay. So what is the CFR? How does the CFR define a finished dosage form? It's a tablet capsule. We'll be talking about it a little in a slide from now, that contains a drug substance generally, but not necessarily in association with one or more other ingredients. I came to appreciate that when I started doing the reviews, and I have a slide typically about a phase 1 manufacturing for a solid oral. Now, throughout this presentation, I'll try to give as much as information as possible, not only from a phase 1 perspective, but also moving on into a phase 2 and a phase 3. It may not be in great detail, but we have the opportunity to talk about those a little after this presentation. The dosage form. One of the primary reasons for this slide is that I needed to have something colorful, since Cecilia had some Cecilia there. But that apart, dosage forms. So what's a dosage form? It's just as a vehicle that delivers the drug to in the body. And there are different types of dosage forms. You have the solid orals, the semi-solids, you have the liquids, gas, and so on and so forth, which are administered primarily through different routes of administration. So you have the ophthalmic route, topical, oral, and then even with the solid orals, if you take the tablet, there is the buccal tablet, the chewable tablets, the immediate release, the modified release, the whole variety. It's just complicated, it's complex, but it's beautiful. So here are the subcategories that we have in the drug product section of an IND. First one is the components. You got to let us know what's the active and what are the inactive ingredients. And one of the things that we find is that in the quantitative composition, we expect the complete composition, the profile of what excipients are being used, and not only the quantity, but also the quality standards. If it's compendial or non-compendial, and we'll talk about the compendial and non-compendial a little down the road. But it's very important for us to know what it is. I'm looking at it and says, okay, it's hypermellose or something. And I know it's probably compendial, but if it's mentioned in the IND, then I know it's a guarantee that it complies with the compendial standard. So we'll appreciate that if you have that full information. Then, of course, we have the manufacturer's name and address, the whole description of the manufacturing and packaging process, which is, of course, will be very graded amount of information that we would expect in an IND based on whether it's in a phase 1 or a phase 2 or a phase 3. Batch analysis. The reason why it's in blue is because of its importance. And we will connect with this with the drug product specification and how batch analysis is important, because this is a great data that tracks the quality of your batch from lot to lot, right from phase 1 to phase 2, to your clinical trial material, registration stability batches, and the post-approval commitment batches. So that's a great way to start there, and also it helps you in setting the specification based on understanding of your process, the formulation, its behavior on storage, and so on and so forth. Analytical methods. I'm not going to be talking about it a whole lot later, but I'll just make a mention that we do recognize when you have a phase 1, you don't know a whole lot about what method to develop, what will be appropriate as stability, indicating capability of the method, and so on and so forth. But we would like to see if it's a small molecule, most likely you're using a reverse phase. So the chromatographic conditions and so on and so forth, if you can provide that, that would be great. But as your drug development progresses, we would expect some kind of a validation data. We don't expect at phase 2 or probably earlier on to have a complete validated method, but eventually it has to go there because this goes hand in hand with your testing of your product. The container closure system, we'll come back to that again to talk about the container closure system. ICH Q6A defines specification as a list of test analytical procedures and acceptance criteria. I'm sure most of you are very familiar with the ICH guidances, and I'll be recommending a few guidances that it's a great read for the weekend. And Cecilia touched upon the stability data, and I'm going to also, we'll come back to stability data, why we need, how much we need, why is it important, and all the other related information with the stability data. Label and environmental assessment. You would think label is not a big deal. It's a CFR requirement. We need the label, irrespective of whether it's a research IND or a commercial IND. Even if there is an approved product being used, because the approved product was used for a particular indication, but the trial, the phase 1 study is probably for some other indication. So we would want the label information. We'll if you've looked at the CFR, you know what I'm talking about. We need the cautionary statement with all the other information that's very relevant as it goes to the clinical trials, the trial site. And then placebo. Placebo can somewhat be challenging depending on what the formulation is, what the nature of the excipients, and so forth, so forth. Now, talking about excipients, they are pharmacologically inactive, but they have to meet the safety profile just as much as the drug substance, except that these don't exert any therapeutic effect. Then the nature of the excipient, the concentration, and the characteristics of the excipients can really influence the performance of the drug product. Either it could be stability or bioavailability and so on and so forth. And one of the things about the excipients is you have a variety of attributes based on which you would choose an excipient for your formulation. So from a standpoint of a regulator, the first thing it should be, it's non-toxic, non-reactive, it's stable. And then of course you have all the other attributes such as patient acceptability, and that comes and kicks in more as the development progresses. Now there are several kinds of excipients with varied functions. I've listed only a part of them, like binders, which achieve the hardness of the tablet. Then you have the lubricants, which are used in manufacturing. Primarily, the excipients can be used as an aid for manufacturing, absorption, or stability, and a different whole range of functions. The one thing I want to mention here is for the multi-dose products, you have the preservative there, and preservative function is very important. And taste masking, where there's the use of the sweeteners for the pediatric formulations. Apart from masking the taste, it also does help with patient compliance. So excipients do play a very significant role with the formulation. In continuing with the excipient, there are a few more slides in the excipients. And from a biologic standpoint of view, there is the protein aggregation, and that's avoided by using some of the surfactants. And then for the osmotic potential and tonicity, even for the parenterals, you use the salts and then sugars. Excipients can serve many functions, as we all are aware of. One classic example would be starch, which can be used in a wet granulation as a paste, but it could also be used as a disintegrant, as a dry powder. Similarly with lactose, which is used as a diluent or a filler in a tablet or a capsule, but could be used as a carrier of the drug powder in an inhalation product. So the same excipient has got different functions, and then also 2 or more excipients may be necessary to obtain the desired property. And this is something probably that evolves as drug development goes by, and then you realize that you may have one excipient is not really doing the function that you are expecting it for, and so you may have to have 2 or more excipients that are compatible. Now, again, coming back to compendial, non-compendial, if it's a compendial, we're good with it. Just make mention of what compendial standards it meets, and then we'll come to the non-compendial in a few slides from now. We talked about the suitability of intended use, functionality. Now, compatibility with drug substance and other components is the key. Like as I mentioned, non-reactive is one of the attributes of an excipient that we'll be looking for. And when you talk about compatibility, it's just not with the APA and the excipient, it's the excipient with the excipient, the excipient with the container closure. And what happens during these interactions is you could have changes in the physical, chemical, microbiological properties, ultimately leading to a decrease in potency or increase in impurities, or maybe even a change in the dissolution profile. Now, in terms of safety and performance issues, suppose you have a preservative which undergoes, it is not stable and undergoes degradation over the period it is stored. It's clearly is an attribute that you don't want to have. It's going to have undesirable effect on the quality of your product. And in terms of safety, the excipients don't have any pharmacological activity, but they're not inert. They do have impurities. Microbial burden is one. I can think of residual solvents. And at least I know of one instance in an Indian application where the manufacturer of the excipient had made a little small tweak to the manufacturing of the excipient, which the NDA applicant wasn't aware of, but it significantly impacted the quality of the product. So even small changes to an excipient can have a very deleterious effect on the quality of the product. One thing I want to point out here is the inactive ingredients database, that FDA website, you can find them. It's pretty useful. Now, it lists all the excipients that have been approved in the currently marketed products. Now, one thing to be aware of is that if just because there is a excipient that's been approved in a marketed product, there is also the route of administration. So if you have an excipient that's been used for oral administration, doesn't mean that it is safe for use in an ophthalmic route of administration. Just want to make sure that if that's the only excipient that you would be able to use, then talk to the agency and see what kind of data you need to support that for the new route of administration. So when you talk about compendial excipients, that's a safe route where we have a compendial reference. It implies that it complies with the compendial standard. Now, when you talk about novel excipients, these are excipients that have never been permitted to be used in a study. So this would require substantial amount of data, especially from a safety point of view. And there is an FDA guidance which talks about qualification of these excipients. The safety is from a standpoint of the dose exposure, the duration of the exposure, and also the route of administration. And CMC does not work in its little hole or its little small world. We interact with the Pharm Tox colleagues to make sure that these are all safe at the level, for the duration, and for the route of administration. From a CMC standpoint, we would require information as much as you would for the API to determine the quality of the excipient that you're proposing to use, which is a new one, is adequate for the proposed formulation and study. So usually it's a type 4 DMF, and we go through the whole thing about manufacture, the specification, the analytical methods, the whole 9 yards as we would for an API. All right, so we have got the formulation ready, so we got to start moving to the manufacturing process. Here I have two comparisons of a small molecule and a biologics, the most simplified version that I can bring up. So in the first one, in the small molecule, this is one is either with the active or with an inactive. I've seen both, where it's blended and then it's just straight filled into the capsule. This is typically in the first in human studies, phase 1, small single dose, those kind of studies. And in the biologics, you have the samples would have to be thawed or maybe pooled, formulated if needed. It goes through sterile filtration and an aseptic fill and lyophilization as required. And then, of course, it has to be reconstituted and diluted as may be the case and before further use. So this is the most simplest form of the manufacturing. It gets complicated. You have a better understanding of your product. You have process controls, more robust process controls as development progresses. What we see in this section is usually a brief description of the manufacturing process and then also a flow chart. And like as I mentioned, the process control depends on the stage of development. And that's understandable. Okay. So going back to the manufacturing, there was a very simple case of where the API was mixed with an excipient. and then filled into the vehicle capsules, and then you would test for assay, microbial limits, and those sort of things before. There's not a whole lot of testing going on there. It's very simple. Now, in this case, there was a capsule which was filled with an API filled with a filler A, as an example, as an excipient. And we tend to look at CMC reviewers would look at the clinical protocol to make sure that the storage statement, the formulation, matches with what's presented in the CMC part of the submission. It was the only instance where I found that the filler in the information presented in the clinical protocol was very different from what was in the CMC section. So the obvious question is, which one, A or B? And so we reached out to the sponsor and have them clarify what it was. So the take-home message here is that please check to see if the CMC section and the clinical protocol, wherever you present these information, are consistent, especially from a formulation and storage condition. We talked about the label, because the storage condition and any special conditions to protect the product from having any deleterious effect of photostability or what have you can be avoided if the label is very clear, and which is why I'm talking about the storage conditions here. Okay, we're moving into drug product specification, which Cecilia also talked about, and there was mentioned earlier also of the test, the analytical procedures and the acceptance criteria in terms of reference to the batch analysis. Why is it so important? The idea is here you set a set of criteria that the product, your drug product, should conform to on its storage. We'll talk about this more as I get into the drug product specification. There are instances where you can include an attribute that serves as a surrogate for performance, and one classic example would be dissolution, either for product performance or from a standpoint of where you're trying to understand the polymorph. And although ICH does talk about it, this is as a last resort. If you have a polymorph issue and it's a low dose drug that you cannot sort out the polymorphs, one way is to look at is to have a discriminating dissolution method to make sure that you'll be able to detect these undesirable polymorphs. We'll make it interactive, a little fun. How many of you are aware of CQAs, critical quality attributes? Okay. So your weekend is all set with ICH Q8. Okay, so this is what Q8 says. It's a physical, chemical, biological, or microbiological property or characteristic that should be within an appropriate limit, range, or distribution to ensure the desired product quality. And this actually shifts from where you are in phase 1 to when you get to phase 2 and you have a deeper understanding of your process, the formulation of the manufacturing process, it shifts. But when from a phase 1, when we look at the IND, from a safety first, it's impurities. The first thing is impurities. Qualification is handled by the form tox colleagues who will look at it and say, where is the tox lot? Where is the clinical lot? Sometimes you don't have a clinical batch manufactured when the IND is in. So you have developmental batches, which kind of can tell us the story about where this is all heading to from a tox lot to a clinical lot. So that degree of confidence is also is very important. So impurities is one, and in case of a sterile product, sterility. Now we often get questions. We are testing for sterility. It's a sterile product. Do we need to test for sterility? What do you think the answer should be? Anyone? Of course, yes. Even for an exploratory IND, there is a very nice guidance on exploratory IND, and if you read it, it will tell you that for micro, if you have a sterile product, it's a sterile product, all right, but you need to test and show that that product that you're going to administer to the patient is actually sterile. So that's very important. And then endotoxin limits is another, that's an attribute that we monitor, and from a biological perspective, it will be immunogenicity and particulate matter. Particulate matter is such a unappreciated attribute, if you will. And I know Maureen is here, and the anti-infective groups is just smothered with lyophilized products, for intravenous administration. And time and again, we see this issue of particulate matter. So the first, and I think there was one slide which talks about is a case study where there was not tested for particulate matter, and we went back to them and said, you need to provide this data to make sure that nothing falls apart. It's one thing is the potency, but the other concern we have, at least from an IND point of view, is the safety of this particulate matter. Okay. Remember, we talked about batch analysis and how do you test those batches? These batches are tested based on the specification you have, and Q6A is very clear about offers a lot of guidance about how do you go about setting a specification. So we talked about the first column is the attribute, the acceptance criteria, and the analytical procedure. So you may have a compendial method. You can indicate those by the appropriate reference there. But if it's an in-house method, you would want to indicate what exactly that method is. This is a cocktail of all the dosage forms. So you can look sterility and then release. So it's just to give a snapshot of what a drug product specification would look like. And irrespective of whether it's a biologic or a small molecule, you would have to know the ID, who is in there, appearance, what do they look like, assay, dose uniformity, release, if it's a solid oral. Impurities, of course, will be definitely part of it. And you can look at Q3B, which sets the tone for how do you set specification, how do you report those specifications, and depending on the nature of the formulation, whether you need to have microbial limits or sterility, water content, and preservative content. So I want to go back to batch analysis to say when you test those batches based on the specification, it gives you a story about where is lot-to-lot consistency. Now we have several years ago, we used to have INDs, which will say in the specification, report results. The batch analysis will say, confirms. Okay, so we have moved on past that, and now we encourage, we realize that it's a phase 1, but it's always good to have some numerical there. So you have something to look forward to say, yes, it's good, or we need to rework, or we need to improve on the process, the formulation, or whatever may be the reason why it's not meeting that acceptance criteria. So our recommendation would be is even when you have one or few batches that you have manufactured, R&D batches, development batches, what have you, you have a feel for what this batch will look like, what kind of quality attributes you're looking for, and set what's called as a tentative specification. It's tentative. And this can be a moving goalpost as you get into the phase 3, where you're more robust, you have a very good understanding of your product, you can start setting specification and then provide a justification as to why the set acceptance criteria is robust enough. And again, this is for a biologic. It's likely different about what you test, but how you test. Methods may be a little different, but again, it goes through the same principles of what attributes are tested: ID, potency, strength, purity, and some of the safety things like endotoxin and sterility. So this is a case study about 3 IV products. All of them are US approved product, FDA approved products. And the intent was to mix all the 3 of them and do a phase 1 study. Right? Well, individually they all look good. They are all approved products. We have the package insert which tells you how you need to do, but how do I know that when you mix all the 3, what happens to the final product? So there was no testing of the final solution, and when you don't have a compatibility demonstrated, it becomes a safety concern. So we reached out to the sponsor and asked them to do some testing, looking for individual, the assay, and the impurities and particulate matter, and how do you accomplish sterility? Now you mix those 3. So that's a frequent question that comes from our microbiology, product quality microbiology colleagues. asking how did you sterilize that? Was the container sterile? Was this sterile? And finally, in these instances, it could be just one batch somebody is doing. All we want is a certificate of analysis, COA. So because that's an authenticated document which somebody testifies saying that these are the tests, this was manufactured by who, when, how much, and everything is good. So I believe none of these case studies that I presented ever went to, these are all real, not made up, and none of them went actually into hold. So it's really gratifying to know that we didn't have to go to that extent because the sponsor was also responding. So we could be proactive in looking at this and identify those issues where it could escalate into a hold issue. We really do appreciate when the sponsors get back in a timely way saying, FDA, here is where what you asked for it, here it is. And so then we can make a decision. Container closure system. I cannot emphasize enough the importance of container closure system. This is in no way less important than the product that gets into this container closure system. So as a package, you have the primary and the secondary packaging. The primary is like a bottle or a vial, which is in direct contact with the product, and the secondary packaging. Now, secondary packaging gets equally important in certain instances because it adds another layer of protection for your drug product over its shelf life. And what we expect in the IND at this stage would be some brief description of the container closure, including if you have a DMF that you want to reference, please provide the letter of authorization. And irrespective of whether it's going to be a DMF for a container closure or for an API, when you submit the LOA, make sure you submit the LOA both to the IND or the NDA and to the DMF. That's needed. So we talked about the last bullet is about the protection and preservation of the drug during the clinical trials. Yes. So if it is photolabile, you have made could be all the protection that you have, or it could be something that you need to protect it from absorbing moisture. It could be a variety of things that could come into play for the secondary packaging. Okay, I think one of Cecilia's slides actually gave the answer to this, but let's see if a show of hands we can talk about how much stability data is required to support an IND. A, B, C, anybody? A. I like that. As a regulator, the more the data, would like it, but we'll take it there and see. So what does the CFR talk about stability? Stability data is required, right, for all phases of the IND. What's very important is that it remains acceptable for the proposed clinical investigation during the study. If the study period is for 6 months or 1 year or what have you, you need data to support that part. If you go with ICH, you can have 12 months, but if your study is only for 1 month, you don't need 12 months data. You don't either have to go through this exercise of generating that kind of data. So all it requires is data for the proposed clinical study duration. Okay, so here it is. What does it do? That data actually supports your study to make sure that it tells us that the product that we made for this particular study will retain its quality. All the attributes will remain unchanged and will remain the same as it was when it was manufactured and you had it tested at release. The one point I wanted to talk about is the degradation pathways of product. What does the degradation pathway of product do? It actually helps to understand or evaluate the intrinsic stability of the API. And it's done on stress condition. We have a lot of ICH guidances on that. And more importantly, it will tell you about the potential degradants that you may see and that you can test that could potentially appear either during the manufacture or the storage. And the other important thing is it also helps to let us know that the method that you're using to do this testing is actually capable of stability indicating. Right there. Okay, so one more question. I don't know how many more I have, but it's, I guess you can like it. So do clinical trial materials have an expiration date? A, B, C? C, maybe. Yeah, that's, I kind of like that. It's no, it leads us to nowhere. And okay, so expiration dating period is not required for the clinical trial material. As long as it meets the standards of your specification that you have tested and it's shown to have not have any changes over the quality attributes toward over the clinical the study duration, there's no requirement. What requires an expiration date, use by date, is for the reconstituted products. When you have a lyophilized product, you reconstitute them, you have to tell us how long you to, how are you going to store it and how long are you going to store it. Because the longer you store, the more the risk. So we'll have more data needed to support that kind of a storage statement. Okay, here is one, I think, we're talking about the particulate matter. This is another one with the lyophilized powder for injection. It has to be constituted with the diluent and then further diluted with the same diluent for infusion. And again, there was no in-use testing for quality assay. Again, come back to particulate matter because we see this oftentimes, even in an NDA application, and which is why I want to really bring up these two case studies to emphasize the importance of this study. Placebo, CFR indicates that for placebo, if you are using a placebo in the study, that we require the information for the placebo, everything about the placebo, the same as you would do for the drug product. And just make sure that you include a test to show the absence of the active, because sometimes, for whatever reason, the active shows up in a placebo, which is not a good thing. Label. The label is such an important thing because when it goes to the clinical trial study, it tells you who made it, when did they made it, what's the address, the lot number. If there has to be a recall or there is an issue in the trial sites, own the responsibility to these clinical trial material. And the patients have to know that this is a product under investigational drug and not something for the treatment. And therefore, and also, you know, the reason why we have an IND in the first place is for interstate commerce to be able to transport drugs across the states. So this is a must for us to have. It doesn't go to clinical hold, absolutely not, but we would want to see, we'll work with the sponsor if they missed submitting something, that we would ask them to submit the asset. Okay, environmental assessment. It's pretty straightforward. All the sponsors would claim an exclusion from environmental assessment. And we talked about the clinical trial supplies. It's the onus is on the trial site for assuring the quality of the records and how they receive it to make sure it has everything that they should be looking for a trial, especially on the labeling part. And they first have to make sure that it is in compliance with 312.6 on the labeling that we just saw an example. Other than that, in this would be all about documentation, the receipt and storage, condition, dosing. And the most important would be the third bullet where it has the date and time, lot number, all the information, including who should be contacted if necessary. Okay, we have developed the product. We're going to submit this IND to FDA. FDA has reviewed this. So there are two outcomes here. No safety concern. So the study it's safe to proceed from all disciplines, including CMC. It's good to go. What the practice we have been following in our branch is looking forward. We want this IND to come back as NDA. So we give forward-looking statements and recommendations saying, hey, here is what you have in the drug product specification. B is something missing here. We can add those things as drug development proceeds. These are all called non-hold comments. The one important message, if anything, that you want to take from this conference is, we want to talk to you as soon as possible, when you're ready. So I think my colleagues, Maureen and Dr. Charu, also emphasize that we approach the right clinical division, because these products can be, depending on the formulation, route of administration, on the clinical indication, it can be very different. What applies to antiviral may not necessarily apply to ophthalmic, may not apply to oncology. So I strongly encourage it when you have these meeting requests, just reach out to the particular clinical division so it's you get the correct response what we can give. So we have these non-hold comments, and one of the things we do is when we have a phase 1, it's looking at it and say, this could potentially go into an end of phase 2. So we request for an end of phase 2. We say, come in for an end of phase 2 or a pre-NDA, whatever works for it. If there is a safety concern, it's placed on hold until it's all satisfactorily resolved. We didn't have very many going on to that. So quickly running through the next few slides, I'm not going to read everything, but the most important point is this: an IND is a continuing process where you can submit an amendment, annual reports. What does it have? Updates, extra characterization data, stability updates. Maybe if there is a change in the manufacturing process or site, maybe it's changed to the synthesis. So those are things that come into the annual report. The one important message is we do review annual reports. We do review amendments. At any point of time, an IND can go on hold if there is a safety concern, irrespective of whether it's a CMC or a form, tox, what have you. Okay, I just needed to have one fancy slide, so and it's getting there. I'm not going to read through this. The most important point is the last big one, meetings with sponsor. There are these milestone meetings which are so very important, both for you and for us. And we would like to see a complete and a good quality submission that we could minimize sending out IR during our reviews, right? And be ongoing with our reviews. So what I encourage you is to come for all, use well these milestone meetings with us. There was one more. Okay, summary. It's quite straightforward that we would need all the information that we talked about to support the ID, quality, purity, and strength of the product. And as we talked about, again, this level of CMC information progressively increases depending on the phase of your trial. I don't think I have a few slides which talks about some of the references to phase 1, phase 2, and the one last thing I want to mention is go back to Q8 because it talks about all the developmental elements as in the pharmaceutical development section. So it's a great read. There's a lot of information that I think will be very beneficial if you can look at it. And I don't have anything. I thank you for your time, and I wish you well in your development programs. Thank you.
All right, thank you so much, Bala and Cecilia. We've got only about 5 minutes. I'm actually going to extend that for an extra 10 minutes. And if you mind, out of a kindness, we have lots and lots and lots of online questions. But you guys can all stick around, hopefully, and pose questions to Cecilia and Bala afterwards. So for right now, I'm just going to go through a whole bunch of online questions. If we could start with the first one, please.
The first online question: if an EU-approved comparator is used in a U.S. study, is reference to the approved EU marketing authorization acceptable in lieu of a CMC section?
Could you please repeat the question?
Yeah.
We were just getting up on the stage. I apologize.
If an EU-approved comparator is used in a U.S. study, is reference to the approved EU marketing authorization acceptable in lieu of a CMC section?
Well, you've heard all day long about it depends.
Case by case.
It's a very profound statement, but it's very if you dwell deep into that, I can say why. We typically would need, if it's an FDA approved product, all we need is a package insert to make sure that this is what will be used. But any of the non-US approved product, depending on the indication, we would need either some minimal information to make sure that those quality standards actually meet if not any better, it's the same as the US approved drugs. If there is an equivalent US approved drug, we prefer that those products be used. But if it's going to be something that's not US approved and it's being brought from outside, we would need some kind of documentation to ensure that that the quality standards meet our expectations.
Next online question, please.
Are accelerated and degradation stability data required for an initial IND for a phase 1 exploratory IND?
We don't expect that at that point, not for a phase 1 now.
Although it may be good information to support the comparability of the toxicology lot of the phase 1.
Next online question, please.
Can you clarify expectations for microbiological testing? In other words, if you are planning to use an alternate method, such as rapid methods, for performing microtesting.
I cannot comment on that. There is an office that specifically reviews the micro in the facility. So.
No, I would recommend just reaching out to the product quality microbiology team, and they would be able to help you answer that question.
Next.
In an initial IND, how much stability data is required for the first clinical if the tox lot does not have stability issues at the proposed storage temperature?
The tox lot does not have stability issues? Could you tell me again the question?
Here you go.
In an initial IND, how much stability data is required for the first clinical lot if the tox lot does not have stability issues at the proposed storage temperature?
So if there is sufficient information to support the comparability of the proposed clinical lot and the tox lot, the stability the stability data from the tox lot could support the stability of the proposed clinical lot during, and the amount of data depends on the duration of the clinical study.
Just if we can just add, the tox lots are generally not as pure as the clinical lots. So to have a good representation of your clinical lot, it's good to have to stand by on its own data. Unlike an NDA where you have supportive stability data, which is what I'm thinking the question is implying, whether we could use that as a supporting stability. I would rather be very hesitant to say yes because of for that reason, because the impurities could be very different. It was a different lot of an API. You could certainly provide all that information, say justify, hey, here is what it is, and it's apple to apple comparison, and then we can take a look at it. But if your duration of the clinical study itself is very short, we don't expect, like as I said, it's not like an ICH recommended testing for real-time and accelerated condition. It's just to support the duration of the study.
Keep going.
Is there any particular requirement in terms of minimum duration for stability data availability for a phase 1 study?
The same. We'll have to repeat it. Please bear with me. It's the same as above. Like we need to have data just to support your duration of the clinical study. That's about it. There's no minimal requirement now.
Okay. Let's do 2 more.
The inactive ingredients database only contains percentages and no reference to the actual approved products. How can we find the approved product information that we can use?
One way you would know is when you send the IND, we would respond saying this is not qualified. Okay. But you can certainly touch base. I guess we have pre-IND meetings that you can always request. We may not be able to offer a face-to-face meeting as much as we like to do that, but we would certainly do have a WRO, which is, anybody take a guess what it is? Written only responses. That's an FDA language. Written only responses. That's very quick, and we should be able to turn around those responses right away.
Okay. One more last question. Make it a really good one there, right?
Are clearance data required for all raw materials used in manufacturing required for an initial phase 1 IND?
We don't require viral clearance for the raw material, but there should be some information to support the safety of those raw materials. The viral clearance is for the manufacturing process of your product.
Okay. Help me thank Cecilia and Balajee for talking to us.
Thank you.